All right, we'll get started. Thank you everyone for joining us. It's a pleasure to have you here. So this is going to be a webinar on leukemia and myeloma. I am Todd Drewley. I'm the Chief Medical Officer from Mission Bio. And we are joined by some distinguished guests today. So we have Professor Hervé Avet-Loiseau, MD, PhD, who's the head of the Laboratory for Genomics and Myeloma at Oncopole in the University Cancer Center of Toulouse, France. We're also joined by Professor Felipe Prósper, MD, PhD, who's the head of Hematology and Cell Therapy at the University of Navarra in Pamplona, Spain. As well as Cedric de Santos, PhD, is the director of AML, MDS, and Multiple Myeloma and Translational Medicine program for Genentech. So thank you, gentlemen, for joining us. I'm going to start by just introducing for our audience anyone who isn't familiar with Mission Bio. So we have developed the only single-cell targeted DNA and surface protein quantitative detection platform that exists in the market today. That platform has been out for roughly 7 years. And most of that time has been spent supporting translational research, and there's dozens of papers that you can find in our library and the website that talk about the utility of single-cell multi-omics for understanding disease mechanisms, therapeutic response, clonal selection, a whole host of different applications. But I think what's exciting for me, I've been with the company for the last 18 months or so, is that there seems to be a real appetite to develop this technology for clinical applications. As a single cell platform, it makes sense to focus on heme malignancies first. And in my prior life as a researcher at Washington University doing genomics research, I was also a customer of Mission Bio, and we talked about doing AML MRD at the single cell level. And that was before we had the ability to also survey surface protein immunophenotyping as well. So it's exciting now to be part of the company and helping bring these technologies into the clinical arena. And we've picked these gentlemen here because they have really been leading the way for these different applications from different perspectives and different global locations. And so it's going to be a real privilege to hear their opinion on these topics. So for anyone listening, if you have questions, we will have a question and answer session at the end of this. We're going to have roughly 45 or so minutes of discussion. And then we'd like to have 10, 15 minutes of question and answers. So if you have questions for the panel, please don't hesitate to drop those into the chat box, and we will get to those at the end of this session. So I'm going to start with Hervé. So you recently received a distinguished award from the International Myeloma Society. That meeting was in Athens, Greece. And despite your assertion to me that that just meant you were getting old and they had to do something for you, I think it really represents the fact that you have done years and years of really high-level work. And the presentation that you gave at that meeting talked a lot about how you see single cell becoming the tool of the future in myeloma research and therapy. So would you mind expanding a little bit on the work that you and your team have done and what you shared at that meeting?
Yes, thank you, Todd. As you know, awards are just for people who are becoming old. So it is just a reason to push me to retire. No. So what we have done for 20 years now is to try to identify the risk factor in myeloma. We know that in myeloma, some patients will have a PFS nowadays about 10 years or even more, but some patients do relapse in less than 18 months. And so it is very important to try to identify these patients, to try to propose to them probably a more aggressive treatment, but more and more now we'll have access to immune therapies, CAR-T, bispecifics, and probably if we are able to identify these patients as soon as the diagnosis time and not wait for the relapse occurring 18 months later, I think it will be a huge progress for the patients. So what we have done, we had last summer in Barcelona international consensus to define the high-risk disease. So now it will be published yearly next year. We do have some factors, but we know that even with that, we do not identify all the patients who will relapse early. And so what we have done is using a single cell technology, and especially Mission Bio, we showed that in about 1/4 of the patient, we're able at the single cell level to identify some subclones, what we call high-risk subclones, with, for example, 17p deletion, 1q gain, 1p32 deletion. And these subclones could be very small, for example, 3, 4, 5% of the cells. So on the bulk analysis, you don't see anything. And what we are, it is in progress. So I don't have the definitive results, but at least in more than 15 patients, we show that these very small subclones are responsible for the relapse which occur 12, 18, 24 months later. And so it is probably, we need to work on more patients, and we have more than 100 patients which are followed now, analyzed with the single cell analysis. But if it is reproducible from patient to patient, I think in the future, we will have to look at that, because we cannot wait for relapse to treat better these patients.
Thank you very much. That makes sense to me as a former oncologist. So I'm going to go ahead and share. I think you have some results coming out in blood. Is this going to be part of that blood manuscript that you expect to see in the near future?
No, this one we did already publish in GCO last year, the preliminary results of that. In blood, we have a paper almost accepted looking at subclones with mutation of the MAP kinase pathway, KRAS, NRAS, BRAF. And we show that in some patients, we have multiple MAP kinase mutations. Most of them are in different subclones, but very interestingly, but I don't have a clear explanation for that. We have some patients who present the two mutations in the same cells. So why tumor cells needs to have a two mutation on the same pathway? It is not published before. We don't have any explanation for that. Probably the cells will take advantage of that. But again, we don't have a clear explanation why we do see that.
So as you mentioned, if you were trying to measure that by bulk analysis, there's no way you'd be able to pick up those clones. Yeah,
no,
absolutely.
Even if we perform whole genome sequencing, I think to look at subclones below 20% of the cells, we don't see anything. So what I'm talking about, it is 5 or even less than 5% of the plasma cells.
Right. Okay. Interesting. And the data I've seen from your team has some of the most rich copy number variants data that I've seen in any disease application on a single cell platform. And, that's been really exciting for me to see how we've been able to take your advice and instruction and try to build panels that include point mutations, copy number variations. And we're starting now to add in surface immunophenotyping and therapeutic targets to that as well. So has that been an advantage in your work to be able to measure copy number variation at the single cell level?
Yeah. As I said before, we can detect 17p deletion, 1q gain, 1p32 deletion. But what we are doing now, we did improve the panel, put in the 3 bispecific or CAR-T targets, BCMA, GPCR5D, and FCRH5. And I think now we are in the time of using immunotherapy for a large number of patients. But we know that it has been published this year in Nature Medicine by my colleague Nizar Badis, that at the time of relapse after bispecific, patients are mostly losing the targets. And we know that with the bispecific, for example, 40% of the patients do not respond to this exceptional therapy. And we don't know why. It could be because of immune dysfunction, but it could be also because the plasma cells already lost the target from this bispecific. And so using a single cell technology, we will be able to look at that and to see if some subclones, even minor, have already lost this target, BCMA, GPCR5D. And for these patients, it will be very important because I don't think it will be the good decision to propose a bispecific or CAR-T to these patients if they have already a subclone which lost the target of the immune therapy.
Absolutely. Thank you. I'm going to shift a little bit to Professor Prósper, who joins us from a car in London. So he's kind enough to join us. I don't think he has video available, but so Dr. Prósper. So I had the pleasure of joining your team in September and seeing some of the early data with the AML single cell MRD results that you've generated. And I think you're going to be sharing some of that data at the ASH meeting in 10 days in San Diego. So would you mind telling us a little bit about the work that you and your team have done around AML MRD with single cell platform?
Sure. Thank you very much for inviting me, Todd. And I'm sorry I cannot connect my video. But anyways, so actually we've been trying to address two main questions with your technology. One is whether this technology was useful to measure minimal residual disease. And actually we selected a very complicated set of samples. Those were cryopreserved mononuclear cells from patients with AML. We had samples at diagnosis, but we actually focus on samples at the stage of minimal residual disease and trying to see whether the technology was able to pick up a minimal residual disease in cases where we also have flow cytometry data, and in some cases an NPM1 RNA. And that was the first question. And I have to say that I have been very surprised with the robustness of the technology, the selection process of the HSPCs, and then even though those were cryopreserved samples, we were able to really pick up some nice signals, some point mutations and specific mutations in individual cells, and to actually see that the technology is really robust in terms of measuring those mutations at the single cell level, even in these samples. That actually trying to measure a number, or let's say to measure the quantity of MRD is something that we're also working, and as you know, we are working with your team to trying to come up with the best way to quantify this MRD to be able to really compare with what we see by fax, with the advantage of the molecular technology in that we really are measuring. When we see the mutation, we really know that there is a cell with a mutation. We don't have to focus on the phenotype of the cell, and that's a clear advantage because it's a more solid and a specific measurement that MRD, where you always have some interpretation one way or another. The second thing is what we have been doing is trying to address whether there is some clonal evolution. And again, with a single cell technology, you don't have to make any assumptions. You see whether a cell has one mutation or two mutations, and whether the same mutation was present at diagnosis and is not present now at the MRD, so that we can really tell what are the clones that are driving the MRD and actually are driving the relapse. When we started doing these assays, we were focusing, as I said, in samples at MRD, but now we have complemented these studies with samples from the same patients at diagnosis, MRD, and then relapse. And that gives us a new picture of what are the clones and what are the mutations that may be driving the progression of the disease, and actually may be very useful in terms of personalized medicine and targeted therapies, because then you can really address those clones that are going to be the ones being responsible for the relapse. So I think there is a lot of work to do yet. Probably the quantification of MRD is one of the things that we've been discussing with you and your team, because that's one of the things that we really need to address. But the potential for clinical application as well is just quite amazing.
Great. I'm glad you feel that way. So one of the things that struck me, and you're mentioning it here, MRD has always been about sensitivity. But as we get more and more sensitive, I think a lot of times we get away from therapeutic guidance. The sensitivity is driven by a patient's own profile of unique mutations. Many of those are not really therapeutic targets, maybe more so in solid tumors than heme malignancies. But at any rate, I think here, we feel like we're on par with the sensitivity of approved assays that are out there. We feel comfortable that we can measure 1 in 10,000. But when I've looked at your data, and conceptually, we're blending flow cytometry with bulk sequencing, but that's been parsed out at the single cell level. And so the numbers are a little bit different than what we've become familiar with from a bulk measure or a flow cytometry measure. And I think that's exciting. We don't know what those thresholds are, and it's early days. So as you say, there's a lot of work to do, but as we get there, I guess my question for you is do you think that the current level of sensitivity is going to be sufficient, because instead you're actually querying therapeutic targets and not just trying to say yes or no, there's disease? You're looking for targetable mutations or protein expression.
Right. I mean, I think that's actually the advantage. You're not only coming back with a number of percentage of MRD or MRD to 1 or to 10 to the 5 and 10 to the 6, but you're actually quantifying specifically the type of mutation you have in the cell. And that is going to give you another option, which is really to target. I mean, some of the mutations are already targeted, IDH mutations. Some of those mutations you can really target. And not only that, you know that some of the mutations are associated with better response to hypomethylating or venetoclax or something like that. And that is also something that we're gonna take the advantage of having that information to really design the therapies of the patients. And even if you have a small clone, you can really monitor that clone through time and see whether that clone is growing and then whether you have to intervene. And those are choices that we didn't have. And when you are complementing the single cell with the antibodies, that is also very attractive in terms of better understanding the disease and better targeting the disease.
Hervé, please.
Felipe, in contrast to you, I am not an expert both in myeloma and AML. So probably my question is not the best one, but one of the problems we have with this technology is that we cannot analyze millions of cells. So to increase the sensitivity for MRD, do you have a pre-program to try to select or to sort by fax the cells of interest before to sequence?
Right. That's what we do. We thaw these mononuclear cells, then we do a selection for CD117, CD34, and then we do the sequencing of these cells. We still have a very large number of cells. That's one of the things that may be confusing at the time of making the percentage or calculating the MRD, and also because some leukemias may not be CD34 positive. So those are things that, as far as I know, also the group of the scientists in Mission B are working and trying to, instead of doing a positive selection, to do some depletion of certain populations, so we can really do not lose any specific potential malignant population. But in any case, this is something that you need to do. You need to do some selection. You cannot sequence 45 million cells. That's not feasible.
Well, Cedric, I haven't forgotten you. And so you sort of sit at the junction of AML and myeloma and MDS from the big pharma perspective. So how do you see pharma adopting, particularly now, it seems like we're in sort of a tough economic time. Is pharma still open to adopting new technologies for these programs? Is there advantages to bringing these precision therapeutics to market with new technologies?
Well, I do not speak on behalf of the CEOs and all the different pharmas, but at least, as you know, from a Roche Genentech standpoint, we do believe in new applications and technologies that can be used in clinical development to inform the therapy, make clinical decisions. And as you pointed out, we have been working with you and Hervé on developing an assay looking at clonal evolution mostly in multiple myeloma. That was the main purpose of this collaboration. And in general, I think from our standpoint, right, at Roche Genentech, we are very keen in developing those kind of assays to enrich and better select the populations that will benefit the most from the different therapies. I think as Hervé pointed out, right, in multiple myeloma, there are point mutations occurring for patients who have been treated with BCMA therapy, and those mutations, which are at the extracellular domain, are disrupting the interaction of the drug with the BCMA. I think with this application, which is combining single cell plus immunophenotyping, you can clearly address those kind of questions in a real-time fashion. And you can certainly, I'm not a clinician, and that was mostly for Hervé and Felipe, but you can think about adapting therapy and switching targets from that aspect. So I think that's a big application where I believe, and I can say that most of the pharma have moved away from the all comers, so one drug fits all patients, into a precision medicine era. And I think that the best example so far in the heme space is very much AML, right, where the disease and patients are very much fragmented based on mutations, as Felipe mentioned. IDH inhibitors and FLT3 inhibitors are currently approved in the frontline setting, and that's the way patients get treated. So this application from Mission Bio, again, combining single-cell DNA and multiomics has a lot of potential from an early disease intervention standpoint, right? I think in myeloma, looking at disease precursor conditions such as smoldering in MGUS patients which have dysregulations of those genes, translocations and mutations. You can also think about the clonal hematopoiesis for MDS, right, where a lot of mutations are predeterminant and mostly will evolve into a full-blown leukemia. So that has application, again, from a diagnostic standpoint, from an MRD to potentially be used as an endpoint, but also to inform and make clinical decisions.
Thank you.
So I have a question for Hervé. Hervé, actually, if you use this technology in a smoldering and even in MGUS, with a copy number variation, would you be able to tell whether plasma cells are malignant or not? Because that's something that I was thinking the other day, because in theory, myeloma or MGUS cells may have some copy number variation that make this different from normal plasma cells. And even though in myeloma you don't have, or you hardly had any normal plasma cells, in MGUS you do, and in smoldering as well. And that's an issue. Well, that's something that I don't know how to address.
What we do is that we have a specific sorter to try to be smooth with the cells. With regular fax, we don't have good results because plasma, especially after therapy, the plasma cells are not in good shape. But we have a specific approach. And so we are sorting first the plasma cells. So of course we have normal and abnormal if you are talking about smoldering myeloma or MGUS. The question now is that we don't know of the prognostic value of a 17p deletion in an MGUS patient. So we need to have more and more analysis like this on single cell to try to have a quite high number of patients followed during time, and to see if an MGUS or smoldering patient with a 17p deletion or 1p deletion will have a much shorter time to progress to overt myeloma or not. so far, we don't have the answer to that, but I think it is a unique opportunity to use this technology to try to address this question.
Yeah, you guys are reading my mind, because my next topic of conversation was going to be precancerous conditions. We're seeing more and more interest in tracking patients with MDS or clonal metaplasia. You've mentioned MGUS or smoldering myeloma. We have publications out there, great work from the lab of Elias Campo in Barcelona, looking at patients that have CLL and looking at the changes at the single cell level that lead to Richter's transformation and high-risk lymphomas. Some of that work has also been done by Dana-Farber and Weill Cornell by Elisa Tenhaken. And then in AML with a precursor condition, Shwachman-Diamond syndrome, looking at the biallelic loss of TP53 at the single cell level. Those are actually the clones that transform into AML. So from my perspective, I see there's a great potential in, sort of monitoring precursor conditions. And as Felipe's asked, do you see that one bad actor that's got a high risk to the patient? And is there something that we could do? Do you see value in this arena for multiple conditions? Felipe, I'll turn it back to you. Yeah.
For sure. I mean, the thing is, those are still very scientific questions. It would be nice to be able to do some of this technology in clonal hematopoiesis in patients with CHIP or with SICUs or IQs, where you can really identify whether those the mutations, the clonal hematopoiesis mutations do evolve during the period of time of following of those patients. But still, the clinical value of that is just very complicated to really anticipate. Those are things that I think we need to do. And the same goes for MDS, for low grade MDS, so low risk MDS, because those are situations in which identifying a specific clones with a specific mutations may actually help us have an idea of what may happen with these patients. But still, we need to accumulate a lot of data and a lot of data, sequential data of these patients to really be able to answer that question. I see a lot of potential, but I still see that we need to do a lot of research in that area. We have also done some studies in lymphoma, in non-Hodgkin's lymphoma, both follicular and DLBCL. And the results, again, are very interesting because we get the cells from the lymph nodes, and we are seeing, again, the same cells with several mutations and the consecutive acquisition of different mutations. So right now, as a scientist, I'm really excited of what I'm seeing with the technology. I'm still not sure what is going to be translatable to clinical application. That's for me the question, and that's what we need to address.
So Cedric, yeah, I wanted to get you also oversee the MDS translational sciences, if I'm not mistaken. So, is there an appetite within Genentech Roche to try to target these MDS or potential MGUS clones?
The simple answer is yes. As Felipe mentioned, we need more data, right? We need to generate more data and evidence to support the claims, but early interventions, I think, is the key theme in oncology these days. And the sooner you can anticipate and prevent the full-blown development, for example, of myeloma or AML, the better it is for patients, right? So I think one of the questions a lot of people have with respect to clonal hematopoiesis is, let's say you have TP53 mutations, but the importance is to quantify how much VEF, right? Because 2% VEF for TP53 is quite different from 20 to 50%. And that goes back to Hervé's initial points around the detection of minor subclones that you're missing with the bug sequencing. And with this technology, you don't miss it. And I think that gives you an advantage compared to the bug sequencing, and you can better pay attention to those particular patients and ultimately, adjust the therapy. So for me and for us in general, I think early disease intervention is the way to go. And I think I will ask Hervé to comment, but there are a lot of trials now in myeloma who are aimed to treat asymptomatic myeloma patients, so SMM and MGUS with different therapies, including immunotherapy. So I think the field is moving, to an earlier detection.
Yeah.
No, yes, I'm not a big fan of these trials in smoldering or even MGUS. The problem is that currently the prognostic model we have in the hands are not perfect.
Yes.
And so we have 20 to 25% of the smoldering patients who are classified high risk who will never evolve to myeloma. So it's difficult to propose a very aggressive treatment to try to cure the patient if some of them will not progress. But you are not in the medical affairs in Roche, but I think, and I am not alone, maybe Felipe will be able to comment. Nowadays, for a company who wants to register a drug for frontline patients, with a median PFS of more than 10 years, it is almost impossible.
Right.
And so it is really a problem for the patients and for the companies. And so I think that, and we have to talk with FDA, EMA, because I think the future will be to dedicate to design trials for high-risk patients, including the patients who have minor subclone who will evolve very quickly. And so if the PFS will be probably less than 3 years, and so it is more reasonable if we want to register a drug than waiting for 10 years, which is totally impossible now.
Fully agree.
Yeah, thank you very much. So one of the things you've mentioned is immunotherapy, and I see an advantage to single cell therapy being you not only have to monitor the disease population, but you can monitor the patient's immune status through some of the immune cells in the same sample. And so, do you see that as an advantage? If we're going to provide a patient with immunotherapy, should we be thinking about T cell exhaustion? Patients who, as Hervé mentioned, respond to bispecifics or don't respond, and we don't know why. I think this is another area where we could do more work as far as what we're seeing in the results of a single cell patient sample. Any thoughts on that from the group?
To me, yes, I can start. I think, one thing, for example, in myeloma that I found missing is looking at the T cell repertoire. So again, Dr. Ballis published some nice data earlier this year suggesting that the T cell repertoire is very much associated with the response to bispecific or T cell engagers. Back in the days during my time at Amgen, those data were published by Monica Brueggemann in BALL. And I think that's, where I view this platform as a single cell, it probably, in myeloma, it can be complementary to the MRD NGS from different vendors, such as Adaptive, where they look at the immunoglobulin rearrangement, and the Mission Bio platform can give you a better understanding with respect to the disease biology by looking at target expression, mutations, and in this case, immune subset from a single cell level. So for me, that's one of the advantages I see.
Mm-hmm. Sorry,
Hervé, I started before you.
No, it's fine. I just have a question for you, Todd. For us in the research lab, the most exciting and what we want and expect is to be able to look at DNA abnormality and RNA in the same cells. So my question is whether in Mission Bio you have some early development or ideas to work on that?
Yeah, this is a great question. It's something that's been talked about for several years, quite frankly. And, I think, we see transcriptome sequencing at the single cell level as an excellent discovery tool. It's been amazing for a lot of, research questions. And so, yeah, you're wanting research questions. I think, you know, where we have, when you try to do cDNA amplification in the droplet, a lot of times the resulting transcript or amplicon, excuse me, the amplicon it can be difficult to distinguish from the DNA amplicon if you're amplifying the same thing. So the design has to be very, very careful. You know, I think it's possible there are people out there that are doing some very, very targeted DNA plus RNA research projects. And that data, I think, will probably come out early in 2024. And so we'll see what that looks like. But it's going to be at a very, very targeted level. And it's right now, it's not something that Mission Bio is going to support commercially. But, you know, I think conceptually it's possible, just requires a lot of design. You know, along those lines, one of the things I'm pretty excited about is DNA methylation. And I will say that we have seen some single cell DNA methylation data that where the DNA methylation itself can act as a proxy of RNA transcription and actually performs quite well when compared head to head with single cell transcriptome data. So, you know, I think that's another possibility to tackle some of these research questions. And we know the SC-TAM-seq protocol that's come out of Barcelona has been very robust as well. So, yeah, it's, you know, I think as we try to focus on clinical applications, developing the, you know, the research output from the platform, you know, those are the trade-offs we're making as a company right now. So, you know, here's the 800-pound gorilla stuff is regulatory questions. So, you know, so it's, and this is particularly relevant because in the last day or two, the FDA has put out comments about limiting LDTs in the United States. Europe did away with LDTs almost 2 years ago now, I guess. And so, you know, there's a lot of pushback. There's a lot of concern if LDTs disappear in the United States. And so this is going to be a pretty healthy and vigorous debate, I think, with the FDA on what they can and cannot do. But I think what the FDA is going to say is Europe.
For non-American, what is LDT?
An LDT is a lab developed test.
Oh, okay. Thank you.
It was similar to IVDD when IVDD was still a thing, but now everything's IVDR. And so we'll get away with a lab developed test, potentially do away with lab developed tests in the United States. But so, you know, from my perspective, where I sit, I work a lot with US researchers and European researchers. You're obviously both European researchers. So I think the Europeans are actually being more aggressive. And I wonder if that's because you have to go, if this is going to come to clinical care, it has to go through IVDR, which is a much longer process. Maybe that's a completely incorrect perception, but I'd welcome your comments, Hervé and Felipe, on what you see as the regulatory landscape for something like this single cell technology.
I don't think so, at least for in my country, in France. For example, we are using NGS on bulk to detect the 17p, 1p deletion, 1q gain, and so on by NGS. It is not approved by anyone. We have our own panel, and it is totally accepted by reimbursed by the government. And so, but probably the law regulation is probably different from country to country. But at least for that, currently, we don't need to have this kind of accreditation.
I didn't realize that.
Right, I agree with Hervé. Actually, in Spain, it's a similar situation. If you demonstrate that a specific test is useful for clinical application, then it will be reimbursed. So we are our government reimburse, the health system reimburse pulse NGS panels for AML, MDS, and it is not for myeloma because we haven't been able to still demonstrate that it's useful and to what level. So they are paying for, and now we really need to move to doing new things and demonstrate clinical usefulness, and then it will be reimbursed. I'm not an expert on regulation, but
yeah, it's probably more difficult in US because the problem is that it is reimbursed by Medicare or by private insurances. And so I don't know, I'm not an expert on that, but it is probably more difficult.
Yeah. So it's really a country by country decision in Europe as to whether or not they will pay for a test if it's been demonstrated to have clinical utility. That's correct.
And I think in the US, it's more about the depth with respect to the analytical and clinical validation of the assay, right? That's really the main differences between the RUO versus the IVD type. That requires a lot of interactions with the FDA, number one, and number two, a more severe and stringent clinical validation of the assay. So I think that's probably the two differences as well.
So do you think this would, the current proposal from the FDA would have any impact on Genentech's appetite for bringing new technologies into their clinical trials, or it really is unrelated?
I do not know, to be honest. I think we'll have to talk to our CDX and regulatory colleagues. That's not my expertise. But what I can tell you is, when you put an assay for a secondary endpoint and using kind of like an IVD assay that requires a lot of validation and back and forth conversation with the authorities, which is quite different from a regular lab-based assay that can be used. So I like the model that Felipe and Hervé mentioned. I think it's all beneficial for patients ultimately. But where you can regulate this and make sure the assay is performing to a certain level that will benefit patients, that's the big question, in my opinion.
But at least for the company, I don't think it is useful because for the trials in the future with the new high-risk definition in myeloma, they will have to perform NGS as they are doing FISH currently, and FISH is not approved by FDA. And so for the trials, you can use which test you want. At least you did validate at least, but you don't need to have IVD or something else.
Yeah.
Agree.
Okay. So maybe let's start, Felipe, with this question. So we also heard the FDA issue a warning about leukemias arising from certain CAR T therapies, particularly those directed against BCMA and CD19. And there was a lot of panic around that announcement in the last few days. Our perception as a single cell technology is that we could identify if there is, insertion in the wrong area or some sort of pre-existing clonal mutation, copy number variation in the CAR T population before it's infused in the patient. So I guess I'm asking the whole panel, what's your take on this sort of worry about leukemias arising from CAR T products? And do you see a benefit to single cell multiomics in being able to mitigate that potential risk?
It has been really, the number of cases is like very, very, very small. It's like one report is 12, another report is 17 cases out of more than 25,000 patients treated. And it's actually T-cell lymphomas. That's what they are claiming. And sure, well, I would say that the risk benefit for CAR T cells is still much in favor of continuing using the CAR T cells. So of course, we have to dwell into what might be the mechanisms, and insertional mutagenesis is the most likely mechanism. And for that, I would say that single cell technologies are going to be useful to at least to understand how the process is, but still it's just too far away from clinical application, because those should be DNA, single cell DNA analysis of like specific areas with new designs, focus on T cell, on different T cells. And I think it's going to be a very useful tool, but for trying to understand the mechanisms of this transformation, similar to what happened with gene therapy in the very beginning of the gene therapy and the development of leukemias, that at the end of the day, we understood that it was insertion in hot spots with specific, I mean, with oncogenes and stuff like that. Now, lentivirus are safer, and we know that it's not going to be the same thing, but we already know that there is some lentivirus still can increase some, some read-through, transcript read-throughs, and some other things that may have something to do with this transformation, but still in a very, very, very limited number of cases.
Thank you very much. Any other thoughts?
I'm not expert of that, and I even didn't know before that we have leukemia with CAR T cells. So,
yeah, okay.
and I will add a few things.
Yeah. Well, it was.
Go ahead.
We've lost Felipe for a moment, so
yep.
Go ahead.
So it's not only the CAR T, right? So Blincyto has published cases reports a few years back that patients actually develop. So Blincyto is an FDA CD3, CD19 bispecific approved for B-ALL, right? And there are some cases where patients experience a lineage switch where they get treated with Blincyto, and ultimately the disease evolves into an AML. And I think from that aspect, your platform can be very useful because you can look at immunophenotyping, where those patients are losing CD19 and other B-cell markers and enrich for CD33, for example. But there are some genetic lesions that are occurring in the co-development of the new AML disease. And the main one is the menin or the MLA translocation. So you can use this single cell platform combining DNA and immunophenotyping to really assess that.
Absolutely. Okay. So before we get to the question and answer session, I have one more question, just throwing this out there to see if anybody has strong thoughts, is the role of artificial intelligence. I get asked about AI probably every other day. And, is it going to make a difference? Is it not going to make a difference? My own personal opinion is that AI is only as good as the information used to train it. But I'm curious from your perspectives, what do you see as the near-term future of AI in this type of work?
Again, I am not an expert of that. Probably it will have, because we have really a huge number of cells. We have a huge number of chromosomal abnormalities. We try to do that just by bioinformatics, which is not perfect yet. I think we need to improve the software to do that. So I don't know if artificial intelligence can have a place. Probably yes, but again, I don't know.
Right. I'm not an expert on that. I wish I were. I knew more, but your concept is right. If we give good information, I'm sure we'll get good answers, but I don't even know how it really works. I keep fighting with our computational biology people. They love that things, the graphic neural networks and all these things. And every time they talk about that, I'm like, okay, I believe you, but it's almost like faith.
Yeah. How do you know it's right? You just sort of accept that it's correct. You don't really know. So.
No, it's a good question, because every day I am talking with our bioinformaticians, and for example, this morning they showed me a patient who said, "Oh, these patients have 5 or 6 subclones." So I went to the patient, and actually one subclone was defined just because the computer did not pick up a 19 trisomy. But when you look carefully at the data, it was just below the cutoff. So it was not a subclone. So we have to be very careful in the analysis of the results we have, and to always ask questions to verify. And again, we are doctors, we are not informaticians, and so it is not that easy.
Absolutely.
Yeah, but the potential, and again, I'm like you guys, I'm not a bio, right? I do spend a lot of time with them and trying to understand, but my view on the AI, it's more like the machine learning and the unsupervised and unbiased way of looking at the data, that they can really reveal, you know, some clusters, and based on the genetic background and the heterogeneity of the disease. But as Hervé and Felipe pointed out, we have to be careful with the interpretation.
Yeah, absolutely. All right, well, we'll move on to questions and answers. I got a question for Cedric. What do advances in single cell technologies enable for drug development that aren't currently possible?
Well,
that's an easy one, right?
Well, I think it's personalized medicine, right? So I think if AML is the best one, myeloma as well, but a lot of patients who get treated with Veneza and IDH inhibitor, at the time of relapse, they do have a lot of MAP kinase mutations popping up. So unfortunately, the MAP kinase experience in clinical trials in heme malignancies has not been very successful. Probably targeting one molecule of the pathway is not sufficient enough. I think there are data from Hervé and Samir Parek combining 2 or 3 different MAP kinase inhibitors. But for me, this platform will potentially allow physicians to intervene and make clinical decisions based on the genetic landscape and the clonal evolution and the emerging mutations. That's one aspect. And the second is also changing the immunotherapy applications if you find that your patients on GPRC5D or BCMA are losing the target. So there is no need to keep treating those patients with those agents.
Yeah. So, and I'll sort of lump multiple questions together. Most people have questions about quote unquote clinical utility, and I think we all do as well. It's a nice platform. The data looks very exciting and interesting and engaging, but how do we know that patients are going to actually benefit if we do this instead of what we've been doing, right? And so we, just as Cedric just mentioned, if you don't see the surface protein, you don't give the drug. That makes sense, but we need to see it in action. So I guess my question, just based on what I'm seeing here, is how much data is going to be convincing to say a single cell technology actually is better for patient care? And it's not going to be for all patients in any situation. It's going to be probably very specific applications for very specific questions. But is this going to be 30 patients worth of data? Is this going to be 100 patients worth of data? Is it going to only be if it's prospective? Can retrospective data suffice? What do you guys think when it comes to clinical utility demonstration?
I can come back on the project we have currently. So we did a single cell analyzed 150 something myeloma patient at diagnosis, all treated in a clinical trial, so the same treatment. And so we found what I call high-risk subclones in about a quarter of them. And so of course, we will follow these patients, but if we show that these subclones are responsible for the relapse of the patient, I think that in the future, we will have to qualify this patient as high risk, as 17p deletion detected on the bulk. And the question now is that will immune therapy will have a better impact on these high-risk patients than traditional chemotherapy we are using now? We don't have the answer, but again, we need to dedicate some trials to answer these questions. And but because currently we say, okay, we detect a subclone with 17p deletion in 10% of the cells. Okay, what to do? We don't have a targeted therapy for 17p, but in the future, I think that we will be able to use CAR T cell directly from frontline therapy. And so I'm not saying that CAR T cells will cure all the patients, but at least we can delay the relapse and the quality of life for these patients.
Thank you.
So
yeah.
in my case, I think we may need not 1 hour, but actually many hours to discuss these things. I think there are two different situations. The application for in AML, I mean, for monitoring MRD, which is, I mean, we are now using MRD for designing the therapies of patients with AML. What we need to do with this technology is to compare this technology with the standard of care, which is FACS probably, at least in general for AML, and see whether there is any advantage in predicting which patients are going to progress early or are going to relapse. And once we can demonstrate that the technology adds significant value to this, would be the time to implement in the clinical application. So getting data both retrospective and using this technology in new clinical trials is what I would like to do. And those clinical trials with this technology as well as FACS will be able to tell us. It's a similar situation is what we are doing in I mean, Hervé knows much more than I do about that, but in myeloma, we use the quantification of MRD as well right now as one of the strongest prognostic factors for myeloma. And we're introducing CTCs in peripheral blood as another factor. And we really need to use these technologies in clinical trials and to demonstrate that they add to the care of the patients, to the prediction of relapse and so forth, and the survival. And then is the time where you can really implement them in clinic. So I don't think it's a too long way to go, but it's some way to go yet.
Yeah. All right. So Cedric, how much does the presence or the absence of demonstrated clinical utility impact your interest, or, you know, say Genentech's broader interest in bringing a technology into a clinical trial? Does it play a role? Does it matter? It must.
Well, ideally, you want to use an assay that will, you know, enrich your population of interest and highlight and enhance the response rate. So it is highly relevant and of interest. That's the simple way to answer. I agree with Felipe. I think it's a combination of retrospective and prospective studies to further validate the platform in comparison to the existing methods and assay. I think, you know, the single-cell MRD assay from the Tapestry in AML is very good in terms of sensitivity, right? 10 to the -4. There will be debate, but I think it's probably 1 log better than the current flow-based assays that most of the people are using. But it has to be compared in a head-to-head fashion or in a retrospective fashion, I think, and which brings a large number of patients need to be analyzed and compared across the different methods. But I think the general question is, how do you foresee using this assay, right? Is it a diagnostic, or is it a monitoring, or it is to inform clinical decisions, or is it a surrogacy? And that's quite difficult with respect to MRD in AML and myeloma, given the authorities. So that really depends on what is the question in the clinical application.
Mm-hmm. Sure. Okay. Well, we've come up on the hour, so I was going to go ahead and close this. Any final comments or thoughts from the panel before we wrap things up?
No, I think it is a great technology. Probably you have some space to continue to improve it, but already now it is really useful, at least for the research, but now I think also for the, I would say, clinical research, working on patient samples and implementing that in trials we are performing, at least in my country.
Yeah, I agree with Hervé. It's great to have these tools. From making scientific questions is amazing, But I know that translation is very important, and I think we're getting to a point where we really need to do trials and to compare these technologies what we already have and see what is the benefit. And we need to do that within the context of clinical trials and specific questions.
Yeah. All right. Well, thank you very much, gentlemen. It's been a pleasure chatting with you. I always learn a lot when I get a chance to talk to you guys. So if I don't see you at the ASH meeting, safe travels, happy holidays. If I do, safe travels, and I look forward to seeing you. But thank you very much for your time.
Thank you very much.
Thank you so much.
All right.
See you in San Diego.
Yeah.
Thank you. Bye-bye.
Bye-bye.